Hello, this is Amy Gines, and this is my final project for genetics. This is sickle cell disease or sickle cell anemia. Sickle cell disease, description and major characteristics. Sickle cell anemia is a genetic disorder which affects hemoglobin and the red blood cells. It's an autosomal recessive disorder, which means that the offspring must inherit two mutant alleles, one from each parent, to inherit the disease and to express it phenotypically. The mutated gene is located on the 11th chromosome, and it's the HBB gene, and it causes, when it mutated, it causes abnormal destruction of the hemoglobin molecule. Red blood cells with mutated hemoglobin are stiff and sticky, and when they release O2, they form a sickle or crescent shape, hence the name sickle cell. The sickle cells have problems carrying oxygen, and due to the sickle structure, they stick to each other and get lodged in small blood vessels, causing many complications. Furthermore, red blood cells usually last up to 120 days, while sickled cells only last approximately 10 to 20 days. So, for the aforementioned reason, individuals with sickle disease anemia usually suffer from anemia, which is the decrease of O2 in the red blood cells. The history of sickle cell disease. Sickle cell disease has been around for at least 5,000 years. It was discovered in 1910 by the cardiologist Dr. James Herrick, and he was the first to notice the shape of red blood cells in patients, described the shape of the red blood cells as having a sickled shape. The prevalence of sickle cell disease. Sickle cell disease affects millions of people worldwide. They're more common in areas such as Africa, Southern Europe, the Middle East, Asia and India, and it also affects at least 100,000 Americans. 1 out of 365 African Americans and 1 out of 16,300 Hispanic Americans. Prevalence of sickle cell disease. You can see there's a correlation between sickle cell allele, right here to the left, and malaria. And the reason why is that there is a heterozygous advantage for which are those who are carriers are protected against malaria in most cases. And these carriers are heterozygous for the allele, which means that they have one mutant allele and one healthy allele. So they do not have sickle cell disease, but they do benefit from having one allele. However, those who have two alleles for mutant alleles and have sickle cell disease also have protection against malaria, but they have all the severe symptoms that come with sickle cell disease. But the reason why this mutant allele has proliferated because of the protective advantage against malaria. The physical effects of sickle cell disease. Because anatomy and physiology are interrelated, the symptoms of sickle cell anemia is caused by a change in the shape of the red blood cell into a sickled or crescent shape. These distorted sickled red blood cells prematurely breaking down results in a constant shortage of O2 in the blood and causes anemia. This also may cause shortness of breath, fatigue, signs of jaundice, and much more. Sickle cell anemia can cause pain and repeated infection when the sickle cells get stuck in the small blood vessels. And the resulting oxygen deprivation of the tissue can cause organ damage. There's been many treatments have been developed. However, most of them only alleviate the symptoms. The only cure is bone marrow or stem cell transplantations. However, both operations are very risky. The molecular cause of sickle cell disease. The molecular cause of sickle cell is a mutation of the HBB gene, which is on the 11th chromosome. The official name of the gene responsible for HBB is Homo sapiens hemoglobin subunit beta, which we can refer to as HBB. Hemoglobin is a protein found within the red blood cells, which helps carry oxygen in the cell. The hemoglobin consists of 4 subunits. 2 of the subunits are called alpha globin, and they're made from the HBA gene. The other 2 subunits are called beta globin, and they're made from the HBB gene. So a normally functioning HBA gene and HBB gene enables the production of normally functioning hemoglobin subunits and the iron molecule within that allows the hemoglobin to carry O2. So when you have a mutated hemoglobin, what you end up getting is a stiff and sticky red blood cell, and it has problems releasing O2. And once it does release O2, it has problems changing conformation shape. So what we end up getting is a red blood cell that ends up looking sickle-celled and shaped. So in summary, HBB allows hemoglobin to normally carry and release O2, change conformation shape so that the red blood cells can bend and go through small blood vessels. And a mutated HBB gene inhibits those types of attributes, which causes all the symptoms that are related to sickle cell disease. So the mode of inheritance for sickle cell disease, sickle cell follows the autosomal recessive inheritance pattern, which means that to receive the sickle cell disease, you must have 2 mutant alleles, one from the mother and one from the father, in order to have sickle cell disease. However, if you only receive 1 mutant allele from one parent and a healthy allele from another parent, you do not have sickle cell disease. Instead, you're just a carrier and are generally considered healthy. As we stated before, the HBB gene is on the 11th chromosome, which is a autosomal chromosome. And this is different from someone who, let's say, gets an inherited disease on a sex chromosome, which those are called sex-linked. So, in summary, sickle cell disease is a recessive inheritance pattern, autosomal recessive inheritance pattern. So now what I like to do is show you 3 different scenarios where I combine Punnett squares and pedigrees. The Punnett square shows you the probability of having a child that will be healthy or have a disorder or just carry a gene or not carry a gene. And inheritance pedigrees are important because they can show the path of passing on these genes from one generation to the next. So it can help you determine who carried a gene in the past or who's going to inherit a gene in the future. So in this scenario 1, what we have is we have 2 parents with sickle cell disease, and then the key what we're going to show is that a mutated HBB gene is a lowercase a, and a normal gene is indicated with uppercase A. So if you look at the Punnett score in the top left-hand corner, you see that both parents have the sickle cell disease, which means that there's a 100% chance that their children are going to have 2 mutant alleles and they're going to have a sickle cell disease. And we can see scenario 1 right here on the inheritance pedigree in the middle. We have Ethan and Ava, and they both have the sickle cell disease and they're divorced. However, before they were divorced, they had a child. And there was a 100% chance that this child named Ebony was going to have sickle cell, and they did. In the second scenario, we have one parent who has sickle cell disease with 2 lowercase a's and one parent who is healthy, who is not a carrier, and they're indicated with 2 a's. And we see here that there is a 100% chance that the child is going to be healthy. They do not have sickle cell. And that's because they will only inherit, there's a 100% chance they only inherit 1 mutant allele. If we look at the pedigree here, to the right, we see Ava and Joe. Ava has the sickle cell disease, 2 mutant alleles. Joe does not. So there's a 100% chance that their daughter, Jenny, is only going to be a carrier, and they're not going to have sickle cell disease. In the third scenario, we have one parent who's a carrier, one only having 1 mutant allele, and one parent who has the disease, which means that they have 2 mutant alleles. And this is very interesting. What we see here is that they have a 50% chance to have a child that has sickle cell with 2 mutant alleles, and a 50% chance to have a child that has only 1 mutant allele, which makes them a carrier. So when we look at the pedigree over here at scenario 3, we see Betty, who is a carrier, and Ethan, who has sickle cell disease. And you can see their children, and it follows the Punnett square that we created, which is they had one child who has sickle cell disease, and they had one child who is only a carrier. And as stated before, there was a 50-50 chance of them having one child who has sickle cell disease and one that is just a carrier. Also, what I like to point out is that if you look at this inheritance pattern, we could also tell that it's an autosomal recessive inheritance pattern, and that it's not sex-linked, because you see the males and the females in equal proportion are both getting this or passing on this gene and inheriting the disease. And it's also you can also see it in most generations. So the options for genetic testing. Genetic testing is required in all 50 states to test for sickle cell disease, and it's either paid by insurance or Medicaid. And if you don't have either, it's still mandatory, and you can still get it at a hospital. It's done by a blood test, and the results usually come back within 24 hours. If you test positive for sickle cell disease during the testing, they'll follow it up with a CBC blood test to confirm the results. Now, prenatal screening is not mandatory, but it's possible, and we have three main options. The first one is CVS, chorionic villus sampling, and that's when they take a sample of the placenta tissue, and then they test it for genetic disorders. And then we also have amniocentesis, which is where they take a sample of the amniotic fluid and they test that for genetic disorders. The con with regards to those two features is that there is a possibility of some type of complication due to the physical invasiveness of the procedure. Now, the third type, non-invasive prenatal testing, is where they just take a sample of the mother's blood, and the mother's blood contains placental DNA, which is analogous with the fetus's DNA. And this is very invasive, and they could use this to test to see if the child has any type of genetic disorders, such as sickle cell. So here we cover the cost of a genetic test, and you see for the newborn screening, it can range anywhere from $15 to $150. It's mandatory, and as stated before, Medicaid or your insurance will cover it. If not, you can still get it done at the hospital, even if you aren't able to pay for it. However, the prenatal testing can be expensive. CVS is approximately $2,400. Amniocentesis is $1,500, and these are usually covered by insurance, but they're not always. And because they're so expensive, a lot of people are invasive. A lot of people are turning to NIPT testing, which can be $200 to $800. So it's less invasive, and it's much more economical. And you could also be able to buy this from commercial companies online. They're not going to be as accurate, but it is a more accessible option for many parents. Now, the genetic testing controversy. As stated before, we mentioned the pros and cons regards to amniocentesis and CVS. They're very invasive, small chance of miscarriage, and they're also expensive if they're not covered by your insurance. NIPT testing is becoming more and more prevalent because it's harmless to the fetus, and the price is relatively low as more companies begin to provide that service. However, it is prone to a lot of false positives, and nobody wants to base early termination of their child based on false results. There's also an issue in regards to privacy. For example, the NCAA test their athletes for sickle cell as of 2012, and people who are or children or teenagers who are kicked off of these teams, it's public knowledge that they have some type of genetic disorder, and there's a lot of stigma that's associated with that. So genetic testing, the results should always be private, in my opinion.